Here comes prophase 1, the longest phase of meiosis. The chromosomes coil and condense to form replicated chromosomes. They are homologous, which means they are similar. There are two homologous pairs of chromosomes per chromosome type, one from the mom and one from the dad. Each pair consists of sister chromatids, which are identical. If crossing over occurs, the two homologous replicated chromosomes will overlap with each other to form a tetrad. Tetra means four, and there are four sister chromosomes, which hence the name tetrad. For the DNA, the number then crossing over may occur where the segments of the sister chromatids will break and will switch and can form and recombine to the different chromosomes. So this is one huge major cause of genetic variation between organisms. In replicated chromosomes, there are twice the number of copies compared to the unreplicated. So this means that there's still 6 chromosomes. There are still 6 chromosomes, though. They don't leave yet. But then metaphase 1 comes. So the square blocks you see are the centrioles. The centrioles create some spindle fibers which latch onto the chromosomes and hold them in place, which prepare them and line them up, so which ensures that when the cell divides, it will have one pair of chromosomes which will, so instead of having one from mom and one from dad, there will be one from mom or one from dad. And these cells that are going to be created are called diploids. The first split splits the pairs from each other. So yeah, and prophase 1's variation comes from the crossing over, but the metaphase 1's variation comes from the choice of the chromosome, because there are so many different combinations and it only adds more. Let's say that one of the chromosomes has red starbursts, dark chocolate, a blue lollipop, and Pepsi, while the other chromosome has yellow starbursts, milk chocolate, cream soda, lollipop, and Coca-Cola. If they both have blindfolds on and they go into a room and then they dump all of their items around their bags, and they can then they have braille dots on each item that show what type of item it is. They will look around and then they'll pick the stuff, and most likely they could have exchanged some things. So one of the chromosomes will have red starbursts, milk chocolate, cream soda, lollipop, and Pepsi, while the other one will have yellow starbursts, dark chocolate, blue lollipop, and Coca-Cola.
Before the start of metaphase 1, crossing over is complete. In metaphase 1, microtubules grow from the centrioles and attach to the centromeres. In chromosomes, tetrads line up with each other in the middle of the cell. The metaphase plate, or the cell equator, in homologous pairs have on one pole and half on the other. In preparation to split, bivalents from either parent can align on either side of the cell. The spindle fibers attach onto the centromeres of each chromosome. Kinetochores, the proteins on the chromatids where the spindle fibers attach of each sister chromatid pair, are turned toward the same pole, causing them to attach to spindle fibers from the same pole. The chromosomes are preparing to split, which will happen in the next phase. During anaphase 1, the spindle fibers begin to pull the homologous chromosomes or tetrads toward opposite poles of the cell. In this phase, unless crossing over occurs, each homologous chromosome consists of 2 copies of the same set of genetic information, so they are haploid. Once this stage is complete, each side of the cell will have 3 pairs of chromosomes, and the cell will get ready to divide. Telophase 1 of meiosis is similar to telophase in mitosis. The chromosomes reach the poles of the cell. The spindle fibers and the centrioles begin to disassemble. Nuclear membranes reform around the 2 new cells, and the chromosomes fray back into chromatin. A crease forms between the 2 cells called the cleavage furrow, and the 2 new cells move in opposite directions. And as the cell membrane pinches off, 2 new daughter cells are formed that are ready to go under meiosis II.
This first split separates the homologous chromosomes in the original chromosome into 2 daughter cells with 2 copies of DNA that are genetically identical unless crossing over occurs. However, another round of cell division is required, so the resulting cell will have only 1 copy of genetic information.
Prophase 2. A second division is necessary in meiosis because it halves the amount of genetic material in each chromosome in each cell. Without this, there would be twice the amount of genetic material needed, which would cause catastrophic problems during fertilization. Anyways, after telophase 1, there is an intermediary stage called interkinesis. During this stage, the 2 cells formed during telophase 1 split, and only after this split, prophase 2 can start. There are 2 stages of meiosis, with prophase 2 starting the second part of meiosis. But there are a few differences between prophase 1 and prophase 2. In prophase 2, the chromosomes are attached to the centrioles by spindles, while in the first stage there are no centrioles or spindles. Also, there is no crossing over between the chromosomes, unlike in the first prophase stage. In addition, prophase 2 has a total of 2 cells originating from the cell at the beginning of prophase 1. In prophase 2, centrioles form and move to the poles of the cell. Also, the spindle fiber apparatus forms and the nuclear envelope dissolves. Only after this, metaphase 2 can start. Metaphase 2. In metaphase 2, each of the daughter cells complete the formation of the spindle apparatus. Single chromosomes align on the equator of the cell, much as chromosomes do in mitosis. This is in contrast to metaphase 1, in which homologous pairs of chromosomes align on the equator of the cell. For each chromosome, the centromere of the sister chromatids face the opposite poles, and each is attached to a microtubule coming from that pole. After this, anaphase stage 2 starts.
In anaphase 2, sister chromatids separate to become daughter chromosomes. Daughter chromosomes are moved toward opposite poles by the spindle fibers. Anaphase 2 differs from anaphase 1 due to the fact that this time, once the cells are split, each new cell contains only 1 set of DNA. During telophase 2, 2 new nuclear membranes form, one around each set of daughter chromosomes. Cytokinesis separates the 2 membranes. Now there are 3 chromosomes found in each cell. By the end of this, there should be 4 cells, and each cell is now haploid. This differs from telophase 1 because by the end, we end up with 4 cells instead of 2. And the final product is ERM.